Hi everyone, my name is Kevin. I'm an incoming third-year PhD student in the Biomedical Engineering Department. I'm in the Systems Pharmacology AI Research Center in Birmingham, Alabama. Go Blazers. Today I'll be talking about PharmAlchemy. So what is PharmAlchemy? It's a database of databases. It integrates drug information, genomic information, disease information, and the goal is to help accelerate precision drug discovery using APIs and LLMs to help collect and mine information. It's supposed to be the starting point for drug discovery, so that's the whole idea. So here's the problem here. So the problem is that there's a need for a comprehensive database that connects drugs, genes, diseases, and clinical trials as a one-stop shop for the starting point for drug discovery. Not only is this important for accelerating precision drug discovery, it's also important for selection and design of clinical trials and patient cohorts, enabling precision medicine, enhancing drug safety, research on drug toxicity, drug safety, off-target effects, as well as inferring relationships between this triangle of genes, diseases, and drugs to uncover hidden mechanisms, disease-to-disease similarities, drug-to-gene interactions, and create new scientific hypotheses. And in addition, we also want to pursue the goal of creating comprehensive reports for drug discovery researchers to conduct target and lead compound optimization, identification, and prioritization. And here's a paper that talks about the importance for combining and integrating different biomedical knowledge bases for repurposing. Here's an overview of the existing data, a sample of some of the data sets that are on the internet. As you can see, a lot of them are not comprehensive, including drug bank. You can get a lot more through downloading instead of getting it online. There's also various interactions that they've tried to combine, but there's no proper schema that integrates everything. There's a lack of clinical trial information. It usually is disparate genes, diseases, or some combination of a pair of the triangle. So here is one of the high-level schemas that we are proposing. It is to integrate drugs, genes, and diseases into some kind of a unified triangle to represent entities and relationships between drugs as R, genes as G, diseases as D. We can create drug sets, gene sets, disease cohorts, and symbolize linkages as if we're trying to create some kind of a biomolecular programming language, R to R, R to R prime, G to G, et cetera, that can create these linkages between drugs, genes, and diseases in order to do queries. And we'll see, today I have a prototype demo that I could show really quickly how this would work. So in terms of how we collected the data from these various sources. Our data set has about 54 million compounds in it, and it has we've collected data with various students from these data sources. A lot of cleaning, harmonization, database staging, putting onto Oracle Apex as kind of like the data warehouse, and developing the prototype in Streamlit, which I will show today. Going more in depth into how we perform the data integration and data harmonization. Data were collected from online databases. We did a lot of conversion, data transformation, data cleaning, and data standardization, formatting, using mesh IDs and different UniPro IDs, et cetera, and making sure that things were properly standardized, UMLS, PubChem CIDs, and combining everything into a final database of databases where each DRG or D2G, D2R, R2G had its own table that were connected using foreign keys. Here's an overview of the data coverage that we have. Like I mentioned, we have 54 million drugs in our data set, but at the center of everything is clinical trials. We could consider clinical trials to actually be a combination of a drug, a disease, and and/or the genes associated with that disease. So it could actually be the superset of drugs, genes, and diseases. And so for a specific entity, which would be that patient, the individual patient. So just to continue on, in order to implement a scientific query using PharmAlchemy, we took the approach of using the funnel and filter strategy. So funnel and filter would mean that we would start with the disease query, perform a genetic gene retrieval to get GET genes, which would be the genetic variants, like from a GWAS database, differentially expressed genes and drug targets, as well as additional genes mined from PubMed abstracts using Google Gemini, perform a linkage from G to G2R using binding DB, and then query similar compounds using the Tanimoto or Jacquard similarity and embeddings for R2R, expanding that to a list of 100 top similar compounds, using Gemini to perform a drug discovery report summarizing the biomolecular properties of all of the top 20 targets, as well as the top compounds from that list. There's also a visualization where we use UMAP with different initializations, spectral, random, and the default parameter in order to perform different visualizations of the R2R or drug-to-drug embeddings. So here are the results of one of the case studies before I jump into the prototype. So the disease input would be Alzheimer's disease. And then we select Alzheimer's disease in the application, retrieve the genes associated with Alzheimer's disease, and then we can individually select genes associated with Alzheimer's disease that we want to consider for funnel, and then that will expand our search using R2R, Tanimoto, and DICE similarities in embeddings, and then we can perform a drug discovery report using Gemini AI to get a comprehensive profiling of the drug target landscape, IC50s, KD predictions, and literature support recommendations for next steps. So in our case study, we found that looking at the CDK5 target for Alzheimer's, we identified 38 potential repurposing candidates with a known protein binding affinity of PKD of more than 5, and identified compounds molecularly similar to known investigational tau and amyloid beta targeting CDK5 inhibitor roscovatine. It also provided recommendations in a drug discovery report encompassing target characterization, compound profiling, and ranking of the most likely repurposing candidates. And then, so right now we are working on a lot of developments for the prototype, integrating with large chemical models, language models, gene embedding models, bioactivity language models like TXGemma, as well as MegaMold-BART, a question answering chat interface linkage to clinical trials data. We have already integrated that into the data set, but we would like to perform queries such as filter candidate compounds currently in phase 3 trials with low toxicity, high protein binding affinity to selected targets. We've also like to perform linkages to jump cell painting, as well as SIGCOMB links, drug gene perturbation, drug cellular morphology perturbation data, as well as integrate validation modules. Validation is very important for this application to be successful. So one of the things that we were working on that I developed was the interval binding attention maps, which is just using ESM2 and cross-attention mechanisms to visualize key residue ligand protein ligand interactions, as well as pharmacological efficacy and toxicity simulations, which is just using a PPI interactome as the JC matrix to perform matrix operations or to simulate drug gene perturbations. And then in conclusion, PharmAlchemy is a knowledge platform that unifies the triangle of drug gene disease relationships into a unified framework for drug repurposing and discovery. It uses multimodal analysis using ETL and embeddings from various sources. I demonstrated that it was used for a scientific case study with Alzheimer's disease, and there's much ongoing work that will potentially position PharmAlchemy soon as a complete one-stop starting point for drug discovery. And then just quickly jumping into the demo, here we are querying for Alzheimer's disease, And then it's going to run a query over pandas GWAS, Rimagene, Gene Expression Omnibus, Open Targets, and PubMed. And then it's going to retrieve a count of how many genes that we've extracted. And then here we have a user interface that shows all the genes that we've extracted, as well as the G2R or the binding DB ligand protein interactions that we can select from. And this is going to allow us to rank by PKD as well as the target. Here we have zoomed in on that table. And also you can click on the DOI as well to get access to the paper, and it's not hallucinated. Here we have selected CDK5 target and its associated linkage ligands. And then we'd like to perform an R2R similarity search. Here we have extracted the top 100 similar compounds using the Tanimoto or Jacquard similarity for Morgan fingerprints, ECP6, I think. And then it has then plotted a UMAP for the queried selected compounds versus the top 100 most similar compounds. And then you can overview the different points in the UMAP plot that tells you the corresponding compound CID for that compound. Here we have zoomed in on one of the R2R embedding graphs, another UMAP, and then we want to generate a drug discovery report. So we use Gemini, and then using Gemini 2.5 Pro, we have created not only a disease section that describes the disease overview, but also describes the top 20 targets associated with that disease, as well as the druggability, feasibility, disease role, pathways involved. We are working on trying to maintain fewer hallucinations, but we have strategies for involving APIs such as Semantic Scholar. Here's profiling of the top compounds that we've found in our top queried and similar search compounds. There's bioactivity, safety profile, novelty, selectivity, mechanism of action. And then here we have the final list of 7 compounds. And then roscobutyne is the top compound from this query. And it also provides key recommendations and future directions for in vitro validation and medicinal chemistry lead optimization. And here's some brief blurb about combinatorial approaches for drug discovery and target biology investigation, and more so studying like the biological mechanism in the context of Alzheimer's disease. Thank you very much. That was my presentation on PharmAlchemy.
Any questions for Kevin? Yeah, feel free to come to the mic, please, and then you might have to turn it on.
Okay. Great talk. So I was wondering, what method did you use to get the embedding for each node?
For each node, okay. So we used Morgan fingerprints from turning Smiles codes for each compound into Morgan fingerprints, and then the embedding. I think I didn't use like a machine learning method. I just I think I just directly used the Morgan fingerprints.
But you have a graph with triplets, right?
Oh, yeah,
with the
UMAP,
relationship, with the link between each nodes. But if you want to get the embedding for all the nodes, Morgan fingerprint is just for drugs or chemicals. What about the diseases? You get the embedding for those too, right?
Oh, for diseases, we would have to use, well, I didn't do embeddings for diseases. I just did embeddings for drugs. But are you asking me what would I do for diseases?
Yeah.
Yeah, so in that case, I would use TX Gemma or I know for genes, there's this SC, there's like a gene to vec, or there's like different gene embedding models, but for disease, I'd probably use TX-Gemma or like one of the more established machine learning models to embed it.
Okay.
Yeah. Yeah.
Thank you.
Yeah.